Okay, so next we have Dr. Ben Dubansky, and he is over at the University of North Texas, and he's going to be talking more about the individual level impacts and ecotoxicology, and then also some of the indirect effects that oil can have on the fish.
Thanks for having me here. All right. So my name is Ben Dubansky, as she said. I'm from the University of North Texas. And I came to the University of North Texas right out of graduate school 3 years ago to work with Warren Berggren, this guy right here, who is kind of an icon in the physiology field, developmental physiology in particular. And a lot of these other people I work with as well. And at the moment, most of the research that we have is actually funded by the Gulf of Mexico Research Initiative and the National Science Foundation, and we're part of the Recover Consortia as well, the Government Consortia. And a lot of the early work I did for my dissertation was with Andrew Whitehead, which Joel had just talked about some of his work too, Fernando Galvez and Charlie Rice. I feel that I should mention these guys as my former mentors, still my mentors. But at the moment, what we're doing is we're working with mahi-mahi, redfish, cobia, and killifish to learn about what the cardiovascular effects of deep water horizon oil spills. And Joel had mentioned about cardiovascular defects in these fish, and the heart starts off as really just a couple rows of cells, and then it turns into a tube like this, and it starts undulating. And this is a mahi-mahi embryo right here. And then as it starts to form chambers, right, and valves, and a lot can go wrong in between the tube and a fully formed heart, which, as you know, is it's a complex organism, and it is the first complex organ. And it's the first organ to form in vertebrates. So this is a killifish embryo, and you can't really tell so much from this picture except for all this red behind here, which is just a bunch of blood pooling. But this is development that has gone wrong. Cardiovascular defects occurred in this fish because it was exposed to oil. And this is development gone horribly wrong, and this embryo is very malformed, cardiovascular defects, and edema in the yolk sac and in the heart chamber as well. But when the oil spill happened, we were very concerned, and we got down there immediately, actually prior to the arrival of the oil. And we were mostly concerned at that point with the estuary itself. As you know, the estuary is where the river meets the sea, and it's a highly productive area, more productive than a rainforest. So what occurred to us was that, as a scientist, it's called a natural experiment. And a natural experiment is not so much that it's natural that we spilled a bunch of oil in the Gulf, but it's natural in the sense that we didn't set up these variables. The variables set themselves up prior to us going out there. So some sites are oiled, some sites were unoiled, and we used that as an experiment. So we went out into the field, and we set traps to catch killifish. And we went into sites that were unoiled and did the same thing. We went all over Barataria Bay. And what happened was, originally we had put out some data about adult fish and the effects, but we were wondering what was happening to the embryos. And what goes on during development when these animals are developing in the marsh that is impacted. So we brought some sediments back from these different sites, and we spawned embryos for these fish, killifish, like taco minnows, or I'm not sure what you call them in Texas, maybe bull minnows or mud minnows. And we put the embryos in the sediment, and what we found was it caused some very severe defects in the fish that were exposed to sediments from oiled sites versus fish that were exposed to sediments from unoiled sites. And what we saw is this, you can see the heart here, it still looks kind of like a tube. Well, as you saw in that first video, it hasn't chamberized properly, and it's stretched out like a long tube. And then we see ascites, which is just fluid that accumulates in the heart, the pericardial area, and in the yolk sac. And what we also found was this complex genomic response. I'm not going to go into it. Actually, I just put this up here to confuse you. So what we know we know a lot about the pathway of detoxification, but we also know that that pathway is interconnected with so many other pathways. It's all this is the pathway that we know well that detoxifies oil. In fact, that pathway is one of the most well-known pathways of all pathways that we know of. It's one of the most best characterized pathways, but still, we know only a little bit about what happens to the animals, what actually is going on inside, especially in the animal that survives, right? So we know the animals are exposed to oil, a portion of them die, but then a portion of them live. And the old way of doing toxicology is simply kill them and count them, right? You figure out how much toxicant it takes to kill the animal. And you mentioned 1 part per billion, and it can be as low as that. Sometimes higher, sometimes lower, depending on the toxicant. It varies. But what you hear from a lot of people was that these Gulf fish have been exposed to oil forever. There are seeps in the Gulf. They've really been exposed for a long time, so they should be able to handle themselves around oil. So we've been looking into this in some very extreme instances, and recently we put out a paper, me and Lauren with Elliot and Cole Matson at Baylor University, and we looked at how animals are able to acquire resistance to toxicants. It's actually very interesting. Now, there's several populations of fish that we know of across the country that have actually acquired resistance to toxicants. They're able to withstand those cardiac defects. That the embryos are just somehow able to survive in water. That if you took a bucket of that water and brought it down the road and stuck some other fish in it, they would just die. And what we do know about this is it was rapidly acquired. So as far as evolutionary time goes, this happened in the last 70, maybe 100 years as we polluted some of these estuaries. Again, it's been repeated. We found it in only a handful of populations. But one thing that's interesting is it's heritable. It seems to pass from generation to generation. And we think it's rare. We think that this situation rarely occurs in nature to this extent. But what we have found is that right here in Texas, there's also a number of populations of killifish that have acquired resistance to the toxic effects of PAH, the toxic chemicals that are in oil. And we found this near the Houston Ship Channel, or rather, Cole Matson found it by the Houston Ship Channel and turned us onto it. So we have a number of these populations, and then we have a number of other populations that we compared them to that were just regular fish, just like your average fish. And we've also compared these to Louisiana fish as well to see the same results. But what you might imagine is that the fish with the best traits survive, right? So this fish is not going to survive. So it's not going to make it. But we also know that there are some trade-offs to this resistance that we found on the East Coast population. There's some researchers that studied these populations on the East Coast that are also resistant to these same type of toxicants, but they found that in the adults they see a lot of cancer, altered immune function. They're very susceptible to disease, as you might imagine, altered immune function. They do really poorly in laboratory conditions. In fact, you take these fish out of the wild and put them in clean water, and they hate it. They want to be in their dirty water, which is interesting. But in the Houston Ship Channel, we haven't found any of that yet. In fact, the fish we found there, we don't see any of this yet. We haven't really looked into it as deeply as we are proposing to. But we have found some interesting things. Of course, they are resistant to cardiac defects that you would expect from oil. They have much higher metabolic rates that consume oxygen more quickly, which is interesting because they have some differences in hypoxia tolerance in some of these populations, and they also have a faster growth rate. When you put these fish in clean water next to some fish from Louisiana or some other fish from clean sites in Texas, they grow much more quickly. And they grow much more quickly. So you see almost, 50 to 70% increase in growth rate in these resistant populations versus, maybe a little under 40% max in some of these other populations. Can benefit for the purposes of this. So we're worried about fishing, worried about shrimping, worried about crabs. We're worried about a lot of stuff. We're worried about what happens on the Gulf, what happened in the Gulf. And there's much aesthetic value in this estuary, and most of you probably feel as strongly as I do about preserving and protecting the Gulf as best we can. And we know that people have lost a lot, and people's lives have been irreversibly changed from the events that took place there. And, you know, we should also mention that, you know, people died on that platform. And that seems not forgotten, but comes and goes away. Again, we found some developmental defects in animals that were exposed to dirty sediments from these oiled sites, but we haven't seen any crazy population effects. In fact, we're seeing some contradictory images of what's going on here. So something that might explain it is, you know, when you have a pair, a mating pair, you have this potential for exponential increase, right? So they mate and they make 2 babies, those babies make babies, those babies make babies, and on and on. Although we have this potential for this exponential increase, we don't really see that. Even though they have this great fertility, they can make so many more offspring, we see that animals exist roughly in a steady state, roughly. Of course, we see these fluctuations like Joel pointed out. And we know that this is mostly because resources are limited, right? So their food is limited, space is limited, predators, whatever. So there's a struggle for existence out there going on that, you know, creates checks and balances that keeps these populations steady. The other thing that we should note is that, you know, organisms are not identical. These are my boys. And there's variation, even in a big population. When I showed you all those embryos, there's variation. They're not all the same. In fact, they're all different, even though they all look the same. And that variation is heritable. And again, those with the best variations are going to survive. This guy is likely not going to survive. However, the ones that do survive in this steady state population that were exposed to oil are likely affected in some way. Some of them are. So right now what we're doing is we're looking at some of the fine physiological features of these fish and their cardiovascular system. So what we want to know is, on the individual level, you know, will or did nature fill in this gap? What did it fill in the gap with? We got this diverse ecosystem like Joel was talking about. We don't really know exactly what happened, but next on the horizon for us is to find out what actually happened to these fish. What about the survivors in this different paradigm of sublethal effects in toxicology? What happens when an animal is exposed to oil and then survives? How does that affect the physiology of the fish? So we're looking at these details of cardiac dysfunction. We're also looking at bird development in the lab that we're working in, and multi-generational studies to see what happens from generation to generation. And this is Dr. Berggren and Hiroshi, who these guys are both bird development specialists, cardiovascular. And just a little teaser on what's going on with birds with something like this, kind of similar to what you see with the fish. We'll go on to questions later. Okay.